Hello and welcome to all my students. So today we are going to do the revision of the chapter Sexual Reproduction in Flowering Plants. I hope you all have attended the lectures, the regular lectures of this chapter, and today I am going to give you a quick recap of this chapter basically from NCERT. Now see students, this chapter is talking about the detailed events what are happening during the process of sexual reproduction in plants. Now, the steps of reproduction you already know. That is, overall sexual reproduction is divided into three events as pre-fertilization, fertilization and post-fertilization, right? Then, coming to pre-fertilization, there are two important events like formation of gametes, then transfer of gametes. Once the successful transfer of gametes takes place, then there is fusion of the gametes to form the zygote. Once fusion takes place, then comes the post-fertilization development, that is formation of the zygote, and then zygote successfully develops into embryo, and from the embryo arises the new offspring, right? So these are the basically events which you already know by the terminologies. However, in this chapter, you have learned the details that how all these steps are taking place in the flowering plant for producing new offsprings, right? So let's start with the ultimate recap of the chapter Sexual Reproduction in Flowering Plants. So now students, here we are going to start first with the first event, that is formation of gamete, correct? Now formation of gamete, basically two gametes you have to frame. One is the male gamete, second is the female gamete, right? So the first step that you are going to study is gametogenesis. Formation of gamete, correct? Now this formation of gamete process, you know beta, it is known as gametogenesis. So basically for reproduction you need two gamete, male gamete and female gamete. So two types of gametogenesis takes place within the flower. So always remember beta, flower is the sex organ of the plant. What is flower? Flower is the sex organ of the plant where all these process of sexual reproduction take place, correct? Now in the flower beta, there are four main whorls, or there are four parts, out of which two are essential whorls and two are non-essential whorls. The two essential whorls are androecium and gynoecium, where the formation of male gamete and female gamete takes place respectively, followed by two more, that is corolla and calyx, which are mainly non-essential whorls as they are not directly involved in the event of sexual reproduction. Rather, they are simply involved in attracting the insects for pollination or protecting the flower in the bud stage, right? So basically, all this event of sexual reproduction is going to take place inside the flower. So you have to remember this point beta first, correct? Now, the two events of gametogenesis are megasporogenesis and microsporogenesis. I hope you all are able to recall microsporogenesis. Beta, mega term we use for female, whereas micro term we use for male. So you have to remember this thing. So wherever I'm talking about megasporogenesis, that means I'm talking about formation of female gamete, and wherever I'm talking about micro, that means I'm talking related to male gamete or male body, correct? Yes. Now see over here beta, in megasporogenesis, the location of megasporogenesis is ovule. So site of megasporogenesis is ovule, whereas for microsporogenesis, the site is anther, correct? Hence, anther is also known as microsporangium, whereas ovule is also known as megasporangium. So now I'm going to show you the structure of an anther, that how male gametes is produced inside the anther, and then I'll show you the structure of ovule, where you will learn how the female gamete is formed. So once the formation of gamete is done, then I'll tell you that how the male gamete are transferred to the female gamete for fusion of gametes to form the zygote. Correct? Starting first with the structure of anther beta. Microsporogenesis is basically formation of male gamete. So I'm writing it again over here. Whereas megasporogenesis is basically formation of female gamete. Correct? Right? Chalo. Proceeding further, beta. Structure of an anther, so that you can remember that basically where this process is going to take place. Now, beta, first of all, anther is the part of male reproductive organ called as stamen. Collective term for stamen is androecium. Now, this is how stamen look like, which consists of two parts: the stalk known as filament, and the two swollen lobes that you can see at the top of the filament, these are known as anther. Now you see how many lobes of anther are there? Two. Then definitely the two lobes are connected also to each other, they are attached to each other. So the structure that keeps the two anther lobes connected is known as connective, right? Now basically I'm going to teach you the structure of anther. Why is anther important for us? Because inside the anther only, beta, male gametes will produce. Inside the anther only microsporogenesis is going to take place. Correct? So for that you have to see the detailed structure of an anther. Here I am showing you mature anther. Correct? The anther which is now ready to produce male gamete. So this is my mature anther. How many lobes you all can see? Two lobes, right? See, this is one lobe and this is another lobe. Correct? They are connected with the help of connective. Now, this whole anther, this mature anther is surrounded by a layer of cell called as epidermis. So, the outermost layer of anther, which is protective in nature, beta, is known as epidermis. So, what am I drawing? This is the outermost single layer of cells, like your skin also have. The first layer is called as epidermis, then followed by dermis, and then so many layers are there, and then finally you lead to blood and lymph and etc. So similarly here also this anther is surrounded by a single layer of cells which is called as yes, very good, it is called as epidermis. Now, epidermis is not the only layer of the anther. Rather, below epidermis, there are three more layers. Correct? So, first, below epidermis, directly, you see a layer of cells called as endothecium. So, this orange color, which you can see on my screen, yes, this is known as endothecium. Now, like beta, epidermis is basically responsible for protection, protecting the underlying cells and everything, right? Now, what is the role of endothecium? Any guesses? If you can remember the class that when ma'am taught you this topic, yes. Endothecium is responsible for the dehiscence of the anther, so that whatever pollen grains are going to be produced inside, they can come out, and then yes, pollen grains, of course correct, carries the male gamete, so that male gametes can also be transferred for fusion. So for that, anther need to burst, and for bursting we need endothecium, which is one of the layer of anther, correct? So basically this endothecium helps in dehiscence of anther, so that pollen grains can come out and they can be dispersed for pollination, correct? Got my point? The two layers of mature anther. Now below this is few layered thick layer called ephemeral layer, middle layer. No, not as such functions are associated with it. And finally, below these middle layer, we have one more layer, very important, which is known as tapetum. Now this layer is important. Why? Because it is the nutritive layer. It provides the nutrition to the developing pollen grains underneath it. Correct? So this is the nourishing layer. Got my point, everyone? So here you see the male structure. This is known as stamen. The collective term for stamen, this is the male reproductive organ, which consists of anther. Anther, the mature anther somewhat looks like this. It is bilobed, connected with the help of sterile band of tissue called as connective. Now each anther is surrounded by 4 wall layers starting from outer to inner. Epidermis, protective in nature. Endothecium helps in dehiscence so that pollen grains can come out, followed by middle layer. As such, it does not have any assigned function and it is very short lived. Its lifespan is very, very less. And then comes the innermost layer, which is known as tapetum. Tapetum is the nourishing layer. It secretes certain nutritive tissues like proteins, amino acids and some trypsin, etc. so that the developing pollen grain can get the nourishment and they develop soon so that pollination can take place. Correct? Right? Now, underneath tapetum beta, there develops chambers called as theca, right? So these are the thecas, these are the chambers also called as microsporangium. Also called as microsporangium. So basically you see anther is bilobed. How many lobes are there? 2. And in 2 lobes, how many chambers, how many microsporangium, how many theca are there? 4. 1, 2, 3, 4. So your anther is bilobed tetrathecus. Write it over here. Mature anther is basically beta bilobed and yes, it's bilobed and tetrathecus. Correct? Got my point, everyone? Okay, so this is how my anther will look like. And now inside these thecas, inside these chambers, what will be produced? With the help of reduction division, pollen grains will be produced. Now the formation of pollen grain with the help of reduction division is known as microsporogenesis. Correct? So thecas, thecas are the location where basically beta microsporogenesis takes place so that pollen grain can be produced. Right? Got my point, everyone? Now, ma'am, you're talking about male gamete, but so far from anther we have reached to pollen grain. Correct. Now, these pollen grains are the main body. These are the haploid male gametophyte inside which beta male gamete formation take place. So far you have seen anther. Inside anther you have seen theca, where reduction division takes place for the formation of pollen grain. Now the anther dehisces from the endothecium point. As a result, all the pollen grains are discharged, but before they discharge, they produce male gamete. Now, next is the structure of pollen grain, so that you can finally reach to right? So that finally you can reach to male gamete. Now, beta, if I talk about the structure of pollen grain, then what is pollen grain? First, you should know pollen grain is a haploid cell, also called as male gametophyte. Why? Because it is a male gamete carrying body. It's a male gamete carrying body. Correct? Now, structurally, beta, if I have to draw the structure of pollen grain, which is very important for you, it consists of two layers. It's basically a spherical body with an average size of 25 to 50 micrometers, and it is surrounded by a two-wall layer. The outer is known as exine and the inner is known as intine. Correct? Now, beta, if you see the exine, it is discontinuous. Correct? It is discontinuous and it's made up of a chemical which is one of the hardest chemical called sporopollenin. So, exine is a discontinuous layer made up of one of the hardest substance called sporopollenin, right? It has some openings through which the pollen tube will come out carrying the male gamete. These openings are, yes, known as germ pores. What are these openings known as? These are known as germ pores. Correct? Now, followed by the inner layer, that is intine. It is a continuous layer. First of all, there are no openings. Second, beta, it is made up of cellulose and pectin. So basically, it is a pectocellulosic layer. Intine and exine, together they are known as sporoderm. Correct? Now, this intine encloses the fluid, and the fluid is known as, yes, you are correct. The fluid is known as cytoplasm, inside which there are two cells. Initially, there are two cells present. The larger called as vegetative cell, right? And the smaller spindle-shaped called as generative cell. Okay, so now beta, these two layers, exine and intine, together, as I told you, is known as germ pore. Now, these are the two cells inside the pollen grain, vegetative cell and generative cell. If shedding occurs in this condition, then it is known as two-cell condition, seen in 60% of the angiospermic families. However, in remaining 40% angiospermic family, beta, this generative cell undergoes mitotic division to produce two male gametes. And if shedding then occurs, then that is known as three-celled condition. So next point you're going to write is pollen grain in 60% of angiospermic families, beta, shedding or dehiscence of pollen grain, dehiscence of pollen grain is seen at two-celled stage. Now, what are those two cells? Yes, generative cell and vegetative cell. Whereas in remaining 40% of angiospermic families, the generative cell further undergoes mitotic division to produce two male gametes. Whereas in 40% of families, dehiscence of pollen grain occurs at three-celled condition. Correct? Got my point? Now, whatever be the condition, ultimately male gametes are produced and your microsporogenesis is complete. Now, one step you have done. Now, let's see how formation of female gamete take place, which is called as, yes, very good. So, it's called as megasporogenesis. Correct? Now, what is megasporogenesis? Where does it take place? It is the method of the formation of female gamete from megaspore mother cell that develops from the ovule, that develops inside the ovule. Now, what is ovule? Where it is present? So, again, going back to the structure of the flower. In structure of the flower, beta, you know that the innermost whorl is gynoecium, which is the collective term for carpel. Typically, a carpel consists of three parts: stigma, style, ovary. The lower swollen base is known as ovary, which consists of ovule that on fertilization transforms into seed. Now, this ovule is the megasporangium where megasporogenesis takes place for the formation of female gamete. So, now I have told you in theoretical manner. Let's see in the diagrammatic manner also, so that entire picture becomes clear in your mind. Correct? So, for megasporogenesis, first you have to see a typical diagram of a carpel. Like there you have seen the structure of a stamen, and here you will learn the structure of the carpel. So, typically a carpel consists of three parts. Correct? This is how the carpel looks like. The landing portion of pollen grains, stigma, the connecting tissue, style, the swollen base, ovary, and this ovary beta consists of ovule, which is our main part. So, since it is a main part, we need to study the structure in detail. So, now here I am going to draw the structure, the most common type of ovule seen in 80% of angiospermic family, that is ovule. This is the ultra microscopic diagram of the ovule. Now let's try to see the parts also like you have seen for anther beta. This is the most common type of ovule beta, which is known as anatropous or inverted ovule. Now, you see there's a slight opening. The opening of the ovule is known as micropyle, right? Then this stalk-like structure which you see is known as funicle, correct? Body of ovule is made up of two layers called as integument, which encloses, yes, very good, encloses a nutritive tissue which is known as nucellus. Now, the base of the ovule which you actually see at the top is known as chalaza. So, this is the typical structure of the most common ovule. Now, why you have seen that ma'am has written over here inverted. Inverted means upside down. So, whatever, if I say that this ovule is kept in an inverted manner, that means what will you consider? You will consider that whatever you are seeing down is actually up, and whatever you are seeing up is actually down. So, what are you seeing down over here? Micropyle. So, that means micropyle is the top portion of the ovule, not the down portion. It's simply because the ovule is kept inverted. That's why you are seeing micropyle at the base and chalaza at the top. But reality is that chalaza is at the base and micropyle is at the top. But in 80% of the families, beta, ovule is kept in this manner only. Hence, we have to draw an inverted ovule. Correct? Got my point, everyone? Okay. Now, beta, what happens? This nucellus is important for the development of female gametophyte. Now, nucellus, which is the nutritive tissue again, like there the nutritive tissue was tapetum, here it is nucellus. So now see beta, what will happen? This nutritive tissue nucellus, now one of the cell of nucellus towards micropylar side right, grows in size and becomes megaspore mother cell and start behaving as megaspore mother cell, right? Now, this megaspore mother cell beta, then it undergoes reduction division, and what will happen to this megaspore mother cell? It will undergo reduction division. Now imagine there's one diploid cell. If it undergoes meiosis, then it will result in the formation of 4 haploid cells here called as megaspores. Correct? Now, from the 4 megaspores, beta, 3 gets degenerated and 1 remain functional. So 3 degenerates, 1 remain functional, and then this one functional megaspore give rise to the entire female gametophyte, female gamete carrying body called as embryo sac. So this one megaspore develops into the female gametophyte. This is the female gametophyte. Correct? If you see over here, then in this diagram, this is the female gametophyte that I have drawn. Yeah. So got my point? This is how the male from this carpal development of female gametophyte take place. Now you have to see the detailed structure of this female gametophyte so that you can understand that where is the female gamete. Now male gamete is ready, waiting. Let's develop the female gamete also, and then once the formation of gamete is done, then we will allow their transfer so that the fusion can take place for the formation of zygote, and then the development of embryo will take place. This is all the events, detailed events that takes place in the sexual reproduction phenomena of flowering plants. Right? Clear? Chalo. Let's see this female gametophyte embryo sac. So how is it developing? It is developing from single megaspore. So now we are going to see the development of female gametophyte. How it is developing? It develops usually, not always, but in 80% of the families, beta, usually it, yes, develops from 1 megaspore. बने कितने थे? बने तो 4 थे, but out of those 4 megaspores, you have seen that 3 got degenerated and only 1 remained functional. That means that 1 functional megaspore is now going to allow the formation of this entire female gametophyte. Correct? So it develops from one megaspore. Correct? Such development is known as polygonum type embryo sac development. Correct? What the terminology is being used? The terminology used for one megaspore developing into the embryo sac is known as polygonum type. ठीक है? याद रखोगे इस चीज को? चलो, very good. Now beta, from this, now how this is going to happen? So I have one megaspore. This one functional megaspore, if you remember the main class, then I told you beta that this functional megaspore undergoes 3 rounds of mitotic generation followed by 7 rounds of mitotic division to give rise to 7-celled 8-nucleate embryo sac. 7-celled 8-nucleate embryo sac, which is actually polygonum, where towards the upper side, now you know ovule is inverted, so even the embryo sac will be inverted, everything inside the ovule will be inverted. So even if you know that chalice is going to be at the base, but here you're going to represent at the top, and micropyle you're going to denote at the lower side. So what will happen? At the top, 3 cells are called as antipodals, right? Followed by 2 central polar nuclei that later fuses. To form a large central diploid cell called secondary cell. So they are going to fuse to form beta large central diploid cell. Correct? And towards the micropyle you have 3 cells, 3 haploid cells. One is your female gamete, for which you are waiting for, and then this female gamete is supported by 2 helper cells also called as synergids. So here goes your main fertilizing body, that is female gamete, nourished by, supported by sister cells also called as helper cells, supporter cells, synergids. Correct? Got my point? So this is the 7-celled 8-nucleate polygonum type of embryo sac, which you see in 80% of angiospermic families. And this is there in our syllabus, so you all should know, remember and learn it. Correct? Got my point? यहाँ तक सबको समझ में आ गया? Now see, your female gamete is over here and pollen grain has already came out. You remember this was my pollen grain carrying the vegetative cell and 2 male gamete if it is 40% of the angiospermic family. Now this is my pollen grain carrying the male gamete. And here is my female gamete waiting for the male gamete to come. So now this transfer of male gamete within the pollen grain to the female gamete for fertilization, this event is known as pollination. So now we are going to study the next, that is pollination scheme. So basically beta, what is pollination? How will you define pollination? It is the transfer of, write the definition portion, transfer of pollen grain from the anther to the stigma of the either from the anther to the stigma of same flower or different flower. Maybe same flower or different flower, maybe born on the same plant body or born on two different plant body, or different flower may be born on same body or different body. Correct? Now, depending upon the type of flower chosen for the transfer of gamete, pollination beta can be of two types: self-pollination or cross-pollination. Self means autogamy, and then cross-pollination means allogamy. Correct? What is autogamy? Transfer of pollen grain within the same flower. Then different flower is cross-pollination. Correct? Got my point, beta? So what is pollination? Pollination is simply the transfer of pollen grain from the anther to the stigma of either same flower or different flower, may be born on the same body or different plant body. Accordingly, we have two types: self-pollination and cross-pollination. Now, cross-pollination, you know, can be further of two types, that is geitonogamy and xenogamy. So what is geitonogamy? How many of you remember? Geitonogamy means transfer of pollen grain within the between the different flower but born on the same body. So when I say same body, that means I am talking about geitonogamy. If I am talking about different body, that means I am talking about xenogamy. Right? Clear? Now, okay. So once beta pollination is done, so basically now question arises, okay ma'am, okay, this is pollination, but what are the agents that are going to help in this process? So depending upon the agents involved for the transfer, we have two types, we have two further categories. So now you are going to see agents of pollination, which can be of two types, either abiotic or biotic agents. Abiotic agents or biotic agent. Right? Clear? Abiotic means what? Wind and water. A means without. Biotic means living organisms. So when living organisms are not involved in the pollination, rather wind or water is involved, then that is known as abiotic pollination. Pollination by wind, the technical term is, you have to remember, anemophily. Whereas for water, the technical term is hydrophily. Correct? Yes or no? Have you all understood till here? Okay, very good. Now, biotic means beta involving living organisms. So when we involve, yes. So when we involve living organisms, like for example, insect, bats, ants, elephant, lizards, snakes, all these animals, when they help in pollination, when they help in the transfer of pollen grain, then that is known as biotic pollination. Right? So depending upon, we have different types of biotic pollination, Like for example, entomophily. It is simply pollination via insects, right? Second is malcophily, pollination by bats. Third, chiropterophily. Sorry, this is bats and just make a slight change. Malcophily means pollination by snails. Ornithophily, pollination by birds. Right? Clear? So these are the different agents of pollination. Now one thing remember beta, if a flower is getting pollinated abiotically, then flower need not to be large, showy, colorful. Even a small inconspicuous flower can be easily pollinated by wind or water. But this is the main condition if the flower is getting pollinated by biotic agents, and plus flower should produce large fragrance, it should produce nectar in large quantity, it should produce pollen grain in large quantity. Why? Because when insects or some living organisms are involved, then in return of pollination, plant need to give certain rewards to those organisms so that they keep on visiting the flower and keep on pollinating. But this reward condition is not applicable over here. Hence, in case of abiotic agents pollinating flower, they are small, inconspicuous, not brightly colored, not producing any fragrance, not producing any nectar. But all these conditions are mandatory for a flower which is getting pollinated by biotic agents. Correct? Understood so far? Okay. Now, next I am going to tell you about the contrivances of pollination, self and cross as a comparison, so that things become more easy for you to remember for long. So now I am going to tell you the contrivance of pollination. Chalo, see. So how many types of pollination do we have? Two. That is autogamy and allogamy. Correct? Now in autogamy, that is self-pollination, the first and foremost condition required is bisexuality. Till the time beta flower is not bisexual, there is no probability of self-pollination. Correct? However, it does not guarantee 100% that okay, अगर flower होगा bisexual तो वो 100% autogamy दिखाएगा, ये assurity नहीं है. But yes, it is one of the prime condition for self-pollination to occur. On the opposite hand, when I talk about allogamy, when I talk about cross-pollination, then the first idea to promote cross-pollination opposite to bisexuality is unisexuality. Right? Now, flower should be either monoecious or dioecious. Correct? Yes. Second is beta homogamy. Homogamy means maturation of the sex organs at the same time. So imagine if your flower is bisexual, it is having both the sex organ, male sex organ and female sex organ, but they are maturing at different time. Male pehle mature, okay, male gamete is released and your female gamete is still not ready. So even after having bisexuality, will your flower show autogamy? No. So this homogamy is very, very important. That is maturation of both the gametes at same time. Correct? On the opposite hand, to promote self cross-pollination, it is opposite to homogamy, that is heterogamy. That is both the sex organs should mature in two different time. Either male gamete should mature first or female gamete should mature first. Third condition which 100% assures self-pollination is cleistogamy, beta. Cleistogamy means flowers which never open. Flowers that never open. This 100% guarantees self-pollination, because if flower is not going to open only, then how the male gametes will move out or how the outside gametes will enter inside. So in such flowers which are cleistogamous, only type of pollination that can occur is autogamy. On the opposite hand is chasmogamy. Chasmogamy means flowers that open. So normally the flowers that open, they can invite the pollen grains from other flowers as well and can help in showing cross pollination, right? So these are certain contrivances. I hope you all have understood this also. Now whatever be the agent, maybe be it abiotic or biotic, ultimately what will happen? Successful pollination ho chuka hai. The transfer of male gamete, that means in short you will say that landing of pollen grain has successfully taken place on the stigma of the carpel. So this was the stigma. Here your pollen grains, that means pollination ho chuka hai and number of pollen grains has successfully landed. Once the successful landing takes place, then starts to assure that whether the right pollen grain has landed, because it is all a random process. Nobody is checking during pollination ki bhai sahi wala pollen grain leke jao. Any pollen grain can land on any stigma. So now this becomes the duty of the stigma to ensure the correct germination of the pollen grain. Hence, as soon as beta pollen grain lands on the stigma, there are some exchange of chemical dialogue between the stigma and the pollen grain, and this is done from the side of stigma to ensure that right pollen grain develops, right? And once the pollen grain passes the test, here come rise to it give rise to pollen tube, which is carrying the vegetative cell that you know and two male gametes behind. And this pollen tube keeps on growing and finally it leads to the ovule, the most common type of ovule, that is polygonum type, right? Clear? Understood everyone so far? If yes, then the next event which is going to take place is, see beta, now what will happen? Finally this pollen tube is going to enter inside the ovule, generally through the micropyle, and now finally both the male gametes will be discharged inside the embryo sac. This is known as pollen pistil interaction. Correct? Now what will happen beta? Suppose this is my embryo sac having antipodals, large diploid central cell, egg and synergids. Pollen tube has successfully brought the two male gametes inside, right? Now these two male gametes, one of the male gamete will fuse with the female gamete to form the zygote. So how zygote is produced? Male gamete fusing with the female gamete, and this process is known as syngamy, whereas the second male gamete fertilizes with the large central secondary cell to produce a triploid tissue called endosperm, right? So now when the second male gamete fuses with the secondary cell or central cell to produce a triploid tissue called endosperm, then this is known as triple fusion. Now, triple fusion, syngamy, together this process is known as double fertilization. क्या कहते हैं इसको? This is known as double fertilization. And double fertilization is a characteristic feature of all angiosperms only. So in whole plant kingdom, the only group to show double fertilization is angiosperm. जैसे ही एक बार ये होगा, soon after this, beta, ovule, transforms into seed and ovary gets the signal to transform into fruit, remaining all other parts get degenerated. Correct? Got my point, everyone? So far, have you all understood? I'm just trying to explain in the minimum possible way. This is just the ultimate recap of the chapter, and I hope you all are understanding. See, this is only possible for those students who have gone through all the lectures or who are having the idea of what is happening in this chapter. So it's just the recap which I'm giving you for this chapter. I hope you all are understanding in the best possible manner, right? Okay. Now, once the fusion is done, now what is post-fertilization event that comprises of first development of zygote, right? So now what is going to happen? Development of zygote takes place. Now there are two types of angiosperms, dicots and monocots. And in both the groups of angiosperm division, the development is slightly different. Here I am going to give you a quick revision of development of zygote in dicots. So see what happens in case of dicots, beta. Zygote ban gaya. Now as soon as the zygote develops, it starts dividing. It undergoes first round of mitotic division, producing two unequal sized cells, the upper called as suspensor and the lower called as embryonal cell, right? Now, this upper suspensor beta further divides to give rise to a 6 to 10 long celled suspensor. Correct? Followed by this lower embryonal cell again undergoes division, two vertical and one horizontal to give rise to 8 celled octant embryo, right? This suspensor beta extracts the nutrition from endosperm, gives to the embryo so that it can further develop. Extracts the nutrition so that this embryo can further develop. Now what will happen, beta? This embryo, the suspensor is giving the nutrition. This embryo will further divide and number of cells will be produced. Such is known as globular embryo. So from octant now give rise to globular embryo. Now this is extracting the nutrition. Further, the shape changes to heart-shaped embryo and finally come rise to the pendant-shaped embryo where all the tissues are very clearly visible. This is the cotyledon of the future embryo, right? This is the radicle which will give rise to the root system protected by the root cap. Radical arises from the hypothetical axis called as hypocotyle. This is plumule, which is later going to give rise to the shoot system that arises from the hypothetical axis called epicotyle. Correct? And finally, this is known as pendant embryo that develops from heart-shaped embryo. Correct? Understood, everyone? Yeah, cool enough. So if you all have understood so much, that means I consider that this is how the zygote of dicot embryo develops. And last is your in monocots. What happens, beta? In monocots, the whole process is same, but the thing which is different is, first over here, this zygote, it undergoes 2 rounds of division producing 3 cells. Like here there are 2 cells, but in monocots it undergoes 2 rounds of division producing 3 cells. And second difference is, in monocot the suspensor does not develop, hence in them endosperm remains intact. Correct? Got my point? Okay, so that's it. I hope you all have understood the entire chapter. Correct? I have given you the major gist of whatever I could have explained. One more thing is left, that is endosperm development. So remember, beta, endosperm development takes place prior to embryo development. It is a nutritive tissue which is responsible for giving nourishment to the developing embryo. This development can be of 2 types, either free nuclear or cellular, right? So just take a quick look from the chapters, from the detailed lectures which have been taught. This was just an ultimate quick recap of the chapter. I hope you all liked it, enjoyed it. If yes, then do not forget to mention in the comment section. And now here goes your homework, where you have to revise the entire chapter from your NCERT, from the lectures, as well as you have to complete your notes. And now, so yes, finally moving to the thank you portion. Thank you for watching the lecture. Hope you all enjoyed it. And like this, we are going to post all the ultimate recaps of your every chapter. Soon we are going to meet with the chapter that is genetics. Stay tuned, stay connected. Thank you for watching me. Bye bye everyone. Take care.